folks, this is chapter 23. We'll be talking about circulation and the circulatory system. So we'll talk about the heart, which is the muscular pump that drives the circulation of blood throughout the body. And we'll talk about how the blood flows through this circuit of vessels to every tissue in the body. And these vessels are the mechanism for delivering oxygen and nutrients to our tissues and picking up wastes and removing wastes from tissues. So the main parts of this chapter are the different types of circulatory systems. We'll talk specifically about the human cardiovascular system and the heart. We'll describe what the structure and function of blood vessels are, and we'll finish up with talking about what the composition of blood is. So there are two main types of circulatory systems. The first basic type is what we call an open circulatory system. So this is in the type of system that many invertebrates like mollusks and arthropods have, and many insects like this grasshopper. So this system is called open because the fluid's pumped through these open-ended vessels and flows out among the tissues. So there's no distinct area, there's no distinction between the circulatory fluid and the interstitial fluid. But in a closed circulatory system, such as in this fish, this is the type of system that squids and octopuses and most vertebrates like us have. And it's called a closed circulatory system because the blood is confined to these vessels, and these vessels keep the blood distinct from the interstitial fluid. And there are three main types of vessels that we'll be talking about. There are arteries, which carry blood away from the heart, and I remember you can kind of think of that as arteries away. So arteries are carrying blood away from the heart, and then veins carry blood back to the heart. And then there are also what we call capillaries. And the capillaries are they can convey the blood between arteries and veins within the tissues. So in this fish, there are two main chambers in the heart. This kind of smaller top chamber being referred to as the atrium, and then in the bottom part is called the ventricle. And we'll see in other organisms like ourselves, we actually have a four-chambered heart. But in this two-chambered heart, we have one atrium and one ventricle. And as I mentioned, these capillaries, these are these small veins that are this network. They're little teeny tiny veins, but they make a massive network within the tissues. And that network we call a capillary bed. And those capillary beds infiltrate all the tissues, and that's where the oxygen and nutrient exchange occurs within the tissues. So in that fish's two-chambered heart, the blood is pumped in a single circulation. So the blood is pumped from the ventricle. It travels to the gill capillaries. The blood pressure then drops considerably, and this allows for the exchange of oxygen back into the blood. So then that oxygen-rich blood is then carried to the body capillaries throughout the body of the fish and back to the heart. But that single circulation, that single circuit, wouldn't be able to apply enough pressure to move blood through the capillaries and the lungs, the capillaries of the lungs, and then throughout the body of a terrestrial vertebrate. So they've evolutionarily adapted to Sorry, there's a distraction there. But they've had an evolutionary adaptation that's resulted in this more rigorous blood flow through this process of double circulation. So they actually have these are again land vertebrates like amphibians and reptiles, and they have this three-chambered heart. So they have a right atrium and a left atrium, and then the ventricle in the center. So their heart is sort of divided into these three main sections. And again, this double circulation, in this process, the blood is pumped a second time after it loses pressure in the lungs. So it's pumped out into the lungs, back to the heart, where it is then pumped out again. There's a second pumping that pumps the blood out into the capillaries of the rest of the tissues. But in all birds and mammals, we have, as I mentioned earlier, we have a four-chambered heart. So we have both a left and right ventricle and a left and right atrium. And the right side of the heart handles only the oxygen poor blood, so that's indicated by the blue, blue vessels. And the left side of the heart handles only the oxygen-rich blood, so the red vessels that you see there, and where those two are exchanging nutrients is indicated in those purple capillaries. So this powerful four-chambered heart is an evolutionary adaptation that supports the high metabolic rates of birds and mammals, and because we use a whole lot more energy than some of the smaller, more less complex organisms. Okay, so moving on to specifically the human cardiovascular system and heart. As I mentioned, we have a four-chambered heart with it's a double circulatory system. So what happens here is we have the pulmonary circuit, so the lungs, the heart and lungs, where the blood is transferred from the heart to the lungs and then back to the heart, and then the systemic circuit. So that's how the blood is pumped from the heart to the rest of the body and then back to the heart. So you can see the pulmonary circuit here indicated in yellow, and then the systemic circuit indicated in green. So this is a pretty simplified image of our circulatory system, just showing, again, this is all the capillaries of the chest and arms, and then down here would be all the capillaries of the abdominal region and the legs. So, again, there's a whole lot more of them than this indicates, but just for simplification purposes. So the general basic flow is, again, this is a double circulatory system. So blood drains into the heart through this, what we call the superior vena cava that flows into the right atrium, and then there's this valve that then allows it into the right ventricle. And then what's draining back to the heart so that's how blood is draining from the capillaries of the chest and head. The capillaries of the abdominal region and the legs is carried back to the heart through this interior vena cava. The blood then is pumped back out of the heart to the lungs through the pulmonary artery. Where it's indicated number 2, the pulmonary arteries carry blood back to the lungs where oxygen exchange occurs. That's where oxygen is picked up back into the bloodstream, and then the oxygen-rich blood is carried back to the left side of the heart. And then that oxygen-rich blood is pumped out of the heart through this large vessel called the aorta. And this is here is an illustration of...
In mammals, blood pumped by the right side of the heart passes through the lungs and enters the left side of the heart. This is the pulmonary circuit. Blood pumped by the left side of the heart is distributed to the other body organs and returns to the right side of the heart. This is the systemic circuit. This pattern, called double circulation, ensures vigorous blood flow to the organs because the heart pumps the blood a second time after it returns from the lungs.
Okay, so when you hear a heartbeat, you hear that gung gung, like a two beats per, when you're counting a pulse. And this is because we have these contraction and relaxation of the heart muscle. So these two kind of sections of the beat are called the diastole and the systole. So the diastole is when blood is flowing from the veins into the heart chambers, and then that contraction is of the systole is when the blood is pushed into the ventricles. And the way that this is controlled, so the heart's pumping, but then it has the contraction that pumps the blood, and then the heart muscle relax. And in order to not let blood flow back into the heart during the relaxation of the muscle, there are these valves that actually close off to prevent the blood from flowing back into the heart. The volume of blood that each ventricle pumps during each minute is referred to as the cardiac output, whereas the heart rate is how many times the heart beats in a minute. Okay, so this is just another image showing how the blood flows through the heart. So we talked about how the blood flows from the capillaries and arteries and veins back to the heart. But it actually flows through the heart as well. So as I mentioned in a previous slide, the blood flows into the heart from the head and body blood vessels through the vena cava, so the superior vena cava at the top of the heart, the inferior vena cava at the bottom, into the right atrium, and it's pumped into the right ventricle. And you can see here this, as I mentioned in the last slide, there's this valve that will shut off, which won't allow the blood to back up into the heart. So from the right ventricle, there's another valve that's open to allow blood out to flow into the pulmonary arteries back to allow the blood to flow to the lungs to pick up oxygen. And then the blood that's flowing back from the lungs flows into the heart through the pulmonary veins back into the left ventricle. Again, we have another valve that shuts off to keep blood from flowing back out. That blood is pumped into the left ventricle and out into the aorta, back out, and to deliver blood, but back out to through the systemic circulatory system. Okay, and as I mentioned, there are those two sections, or those two parts of the heartbeat, the diastole, and again, this is when the heart is relaxed and these valves are closed. And this is when the blood is flowing into all four of its chambers. So the blood enters the right atrium from the vena cava and the left atrium from the pulmonary veins. So blood is flowing into the heart both from the lungs and from the rest of the body. So the valves into the lower portions of the heart are open. The ones flowing out of the heart are closed. The second part, systole, begins with a very brief contraction of the atria that completely fills the ventricles with blood. Then the ventricles contract, and the force of their contraction closes the AV valves and pumps the blood into the large arteries. So blood flows into the relaxed atria during the second part of systole, and as indicated by these green arrows here. Okay, so this contraction and relaxation of each chamber of the heart has to follow a pretty precise sequence and timing to pump the blood through the body. So this is an involuntary muscle contraction. It's not like our muscle cells, which respond to signals from our brain. This is the cardiac muscle contracts on its own without any signal from the nervous system. So these have to, as I mentioned, have to be pretty coordinated. So how does that coordination happen? So there's this group of cells that make up what's called the sinoatrial node. So we refer to that as the SA node. It's also commonly called the pacemaker of the heart. So these are the cells that dictate the pace of the pulsing of the heart. So what happens here is there's signals from the SA node spread quickly through both atria, making them contract in unison. So the right atrium and the left atrium contract in unison. And those impulses pass to a relay point called the AV node, or the atrioventricular node, which is called AV node, that's located between the right atrium and the right ventricle. So this AV node delays the signal for about a 0.1 second, which is the time it takes to empty the atria completely before the ventricles contract. And then these specialized muscle fibers that are indicated in the orange sections here relay signals to the apex of the heart and then up through the walls of the ventricles that triggers a strong contraction that drives the blood out of the heart. And that stent is actually like a mesh tube that keeps the artery open so that the blood flow can be increased moving through it. Okay, so moving on to the structure and function of blood vessels. As we've talked about throughout all of Biology 101 and 102, how structure and function work together, the structure of blood vessels fits their function. So again, we have arteries. Arteries away, they carry the blood away from the heart, and these have elastic fibers that recoil and stretch. They have a thick layer of smooth muscle in their walls that can constrict to reduce blood flow should it need to be constricted, and can relax to expand blood flow. Veins have these thin inner walls and low, because they have lower blood pressure and lower velocity. They allow blood flow towards the heart through these thin inner walls. And then capillaries, as I mentioned, this is where all that oxygen exchange takes place. They have very thin walls. So those thin walls, the gas exchange can actually happen across their very thin walls, their very thin epithelial tissue. And this is just an actual micrograph image of a capillary embedded in smooth muscle tissue. It's kind of an interesting picture. Okay, so this image just illustrates the route that molecules take in diffusing from blood and capillary tissues into the cells. So the cells are immersed in this interstitial fluid. So these are the tissue cells at the bottom here. And molecules like oxygen and nutrients, so these oxygen are like the little gas bubbles are in white, and the nutrients are in red. And they diffuse out of the capillary into the interstitial fluid, and then from the fluid into the tissue cell. So this is how the oxygen and nutrients, you can see it, they flow right out of those thin walls of the capillaries, and then can be taken up through the cell membrane. And we talked in BIOL 101 about how cell membranes allow for the passage of gases and nutrients across the cell membrane. Okay, so we talked about the diastolic part of the heartbeat and the systolic part of heartbeat. This is also how we measure something called blood pressure. And blood pressure is a way to measure the cardiac output and the resistance of the the vessels. So blood pressure, as I mentioned, is highest in arteries as they're being pumped away from the heart because it's closest to where the actual pumping occurs. And it's slowest in the capillaries. So it slows down in these small capillaries, and that's again where the gas and nutrient exchange is taking place. And one way blood keeps moving through hearts is by the use of these both valves and contracting skeletal muscle tissue. So we have contracting skeletal muscles that are helping to continually keep our veins moving blood throughout, and then these valves that again close off in order to keep blood from flowing backwards. So I mentioned blood pressure. So again, this is measured. It's a measure of what the cardiac output of the heart is, and it's measured by those systolic and diastolic pressures. I'm going to walk through an image in a second on how that's actually measured. So the reason we measure this is it's an indication of whether or not that you have a disease called hypertension. Hypertension is also referred to as high blood pressure. And this is a serious cardiovascular problem, and it can be controlled through diet and exercise and often medication. But it's if you have it and don't treat it and don't pay attention to it, it can cause major catastrophic events like heart attack and heart disease and strokes. And this is how you probably have all been to the doctor and have had them put this cuff on your arm to measure your blood pressure. I'm sure you knew that's what they were measuring, but this is actually what takes place during that process. So the cuff's wrapped around the upper arm where large arteries are accessible, and the cuff is inflated till it's strong enough to close off the artery and cut off blood flow to the lower arm. And then a stethoscope is traditionally, I know they have some automated like digital versions now, but the stethoscope's used to listen for vibrations of blood flow in the constricted artery wall as the cuff is gradually deflated. And the first sound of blood spurting through the artery indicates that the pressure exerted by the cuff has fallen below the pressure in the artery. And the pressure at this point, that's what's referred to as the systolic blood pressure. And then the sound of the blood flowing unevenly through the artery continues until the pressure of the cuff falls below the pressure of the artery during diastole. And now the blood is flowing continuously through the artery and the sound of the blood flow ceases. And that's when the second reading is taken, and that's the diastolic pressure. These are the ranges of where you want your blood pressure to be. So you typically 120 over 80. So the top number, the first reading being at 120 or below, and then the diastolic being 80 or below is the normal healthy range. If you're somewhere a little elevated, you may be what they refer to as pre-hypertension, where a physician might say, make sure you keep checking your blood pressure regularly because you're at risk of reaching the condition of hypertension. And stage 1 hypertension is in the 140 to 159 over 90 to 99. And then if you're above those numbers, you're at pretty high risk of heart disease. So, as I mentioned, muscular contraction helps blood flow through our veins and arteries. But we also have these capillary sphincters, which are able to open and close and allow blood flow to the capillary beds in particular reasons. And these can function by opening and closing to increase or decrease blood flow. And as we've talked about, the capillaries are where the transfer of substances occur, and it occurs again right through the capillary walls. The capillaries are the only blood vessels with walls that are thin enough for substances to cross. And blood pressure is what forces fluid and small solutes out of the capillary out of the capillaries. So and osmotic pressure, so we've talked about, if you remember what osmosis is, so flowing from an area of high concentration to an area of low concentration. So osmotic pressure draws fluid into the capillary because the blood has a higher concentration of solutes than the interstitial fluid. So the blood is highly concentrated in solutes, so water essentially will be wanting to flow back into the capillaries. Okay, so finally, what does blood look like? What is blood made of? So blood's composed of two major components. There's the plasma, or the liquid components, and then there's the cellular component. So if you take blood and you put it in a centrifuge and spin it really fast, it will actually separate out and look like this, where the plasma is sitting on top and the cellular components are in sort of a pellet at the bottom of the test tube. So the plasma is it's about half and half. There's a little bit more plasma than there are cellular components. It's mostly water. There are some dissolved ions and proteins and nutrients included in the plasma. The cellular components are a couple of different types. So we have red blood cells, and these are what transport oxygen through that protein that we talked about called hemoglobin. There are white blood cells, and these are also called leukocytes. Word leukocytes. So we're going to talk about leukemia being a blood cancer because it affects these white blood cells called leukocytes. And these are our defense and immunity. These are the guys that fight our infections. And some of them are also able to actually engulf and digest bacteria and things. And then we also have platelets. And these are cell fragments that are actually what make our blood clot. But so they're the components, they're cellular components that bind together to form a blood clot on injured tissues. So what happens, we have this hormone Erythropoietin, EPO, and this stimulates the bone marrow to produce red blood cells. And if we don't have enough red blood cells, these are essential for a healthy functioning of a body. If we don't have enough, we have this condition called anemia. And what happens with anemia, because red blood cells are the component of blood that deliver oxygen to our organs, this causes a lack of oxygen in our tissues and it can lead to serious fatigue and other types of lack of functions. So what happens, I mentioned that there are platelets which are important in blood clotting. So blood clots are essentially plugging a leak when a blood vessel is injured. So blood contains this self-sealing material, those platelets. They adhere to connective tissue of damaged vessels and help convert fibrinogen to fibrin, that's what actually forms the clot. So again, here we have the platelets that are adhering to the exposed connective tissue, so that the lining of the blood vessel is damaged and the connective tissue in the vessel wall is exposed to the blood. So those platelets, the purple guys there, rapidly adhere to the exposed tissue and release chemicals that make the nearby platelets sticky. So soon a cluster of the sticky platelets form a plug that quickly provides protection against additional blood clots. And then these clotting factors in the plasma are released, and clumped platelets set off a chain of reactions that amplify that response. So this is, if you remember, we talked about that idea of positive feedback. This is one of those situations where the body's not undergoing negative feedback, but this is positive feedback. So the reaction is driven forward in order to create the blood clot and form that plug. And then finally, these threads in here are what I referred to as fibrin. And that fibrin actually helps to kind of bind things in place and form the clot. Okay, and finally, I mentioned that there is a disease called leukemia. Remember, those leukocytes are the white blood cells. So leukemia is an example of a blood cancer where it's a cancer of those leukocytes. So there's a lot of study, probably heard in the news, the controversy and discussions around using stem cells for treating all different types of disorders and diseases. And what this is, stem cells are they're kind of unspecialized cells that are made in the bone marrow, and they can, as they divide, they can give rise to a couple different types of two different types of cells. So they can create these lymphoid stem cells or myeloid stem cells, and those lymphoid stem cells will reproduce and divide to create lymphocytes. And those lymphocytes function in our immune system. But the myeloid stem cells can differentiate into other white blood cells and platelets and erythrocytes, and those will continually produce and replace all the blood cells that are needed throughout life. So this example of leukemia as being a cancer of the white blood cells, and that's because cancerous cells will grow uncontrollably. So a person with leukemia has an unusually high number of leukocytes, but they're defective. So they're reproducing rapidly, but they're not functioning properly. So in this situation, These stem cells can be used to reproduce healthy leukocytes and actually transplant those leukocytes into a person with leukemia and eventually replace all of the cancer cells. And that is all that I have for this chapter. I will note that a test is coming up, so keep your eyes out for the announcements, and I hope everybody's staying safe and have a great day.